Hello students, I am Dr. V. Sudarshan Rao. The module that we are going to learn today is biological hazards. It's a part of food safety and quality control. More than 200 different types of diseases are spread through consumption of unsafe food. So, it is in this context it is important to understand what are those food hazards. Food hazards can be classified into three different types: physical, biological and chemical hazards. Among them, biological hazards are very important because they cause lot of acute foodborne illnesses and also economic loss in terms of health burden on the country. In this context, it is important to understand that how to control these biological hazards during food processing to ensure food safety. Learning objectives for this module are the type of biological hazards that they come in food, and what is the nature of biological hazards, and what are the sources from which foods or which place these things are coming into food, and then finally health effects, what does it do to you, to the consumer, and their control, how do they control these biological hazards. These are three learning objectives for this module. So, before we go into the actual topic, we try to understand what is a biological hazard. Any biological substance in or condition of food with the potential to cause an adverse health in humans is called biological hazard. So, what are the types of biological hazards? The most common biological hazards are categorized into the bacteria, virus, fungi, protozoa, helminthes, algae. Now, we try to see each one of them. So, what is bacteria? Bacteria is a microscopic living organisms. They lack distinct nucleus and omnipresent. So, examples of bacteria could be E. coli, Salmonella, Staphylococcus aureus, Bacillus, etc. Then comes to the virus. The virus are ultra microscopic organisms which require host for multiplication. That is very important. They can't do on their own, and they contain either RNA or DNA. Examples are hepatitis virus, rotavirus, Norwalk virus, etc. The third one is the fungi. These are all actually the organisms which are saprophytes. They lack chlorophyll. They actually depend on the other organisms, and examples of them could be Aspergillus, Penicillium. So, these are some of the fungi that you find in food. Then the fourth one is called protozoa. These are unicellular eukaryotic organisms. The examples could be Toxoplasma godini, and then helminthes are the parasitic multicellular organisms, and they also could be present in foods in like meat and the other things. The last one is algae. Actually, these are the toxic organisms which could be, some of them are toxic. They are large group of eukaryotic organisms, unicellular, to some of them are multicellular, some of them are microscopic unicellular. Their examples could be blue green algae, dinophysis, etc. Now, we will try to understand each one of them. First, we go into the E. coli. These are some of the E. coli are pathogenic, that we are trying to understand what are those things which are pathogenic. E. coli are the gram negative non-sporing rods, motile with peritrichous flagella. The most of the strains that we have in our own intestine are harmless, but some of them are pathogenic. What we are trying to understand here is the one which are pathogenic. So, they are present in guts of humans, even animals. Its presence in food is considered as a fecal contamination of food or water. Some of the E. coli strains are pathogenic and cause traveler's diarrhea, hemorrhagic colitis, hemolytic uremic syndrome, etc. Based on their pathogenicity, E. coli are classified into 5 different strains. The one is enteropathogenic E. coli, then we have enterotoxigenic E. coli, then enteroinvasive E. coli, enterohemorrhagic E. coli, and enteroaggregative E. coli. Based on the nomenclature itself, we try to understand how they are actually causing any pathogenicity. The type of diseases vary with different virulence factors, hemolysin production, toxin production exhibited or expressed by the organism. That is how these 5 are classified. A wide variety of foods have been implicated in, including milk, milk-based sweets, non-vegetarian foods like meat and fish. These things are generally implicated in foodborne diseases where E. coli is there. In India, E. coli 0157H7 was found in beef, milk, paneer, ice cream. but in UK it is more common in beef burgers, meat and dairy products. The control of E. coli in food production is done through effective control of raw materials, process condition, post-process contamination. The next organism that is the most important one is the Salmonella. This is a facultative anaerobic, usually motile. It's a gram-negative rod. It causes severe food poisoning, typhoid and paratyphoid. The internationally the most dreaded organism is called Salmonella because it causes more foodborne illnesses throughout the world. It's mainly found in raw meats, poultry, dairy products. In India, many other products have been implicated in outbreaks like kheer, fish, stale rice, frozen food, etc. In India, Salmonella was found to be more common in meat, poultry, seafood and koa. Salmonella can cause disease even if it is in small number. Sometimes they say that even one organism can cause foodborne illness. So that's the reason why this is considered to be a zero tolerance organism. Young children, immune compromised people, sensitive individuals and elderly people are much more likely to become ill after consumption of low number of cells because the pathogenicity is very high. The incubation time is 12 to 72 hours because the incubation time here what we mean is that after consumption of a food which is contaminated with Salmonella, the first symptom may appear after 12 hours and it can go up to 72 hours. The main symptoms, what are the main symptoms of this salmonellosis is the diarrhea, abdominal pain, vomiting and fever. But if the organism is typhoid, it causes typhoid fever. So here also how do we control this organism? It's raw materials, personal hygiene, equipment, environmental hygiene, manufacturing process conditions, post-process contamination, retail and catering practice, all those things, even consumer handling also can contaminate the foods. That's why essentially it is hygiene that we are talking. The next organism that is pathogenic organism is the Listeria monocytogenes. It's a gram-positive foodborne pathogen. It causes the disease called listeriosis. It's widely distributed in the environment, occurs in almost all food raw materials. There are 6 distinguishable species of Listeria that are recognized. The monocytogenes is the most common: Inacula, Welchmeri, Siligeri, Innovi and Greyi. These are the 6 different types of species that have been identified. The most commonly occurring species in food are Inacula and monocytogenes. In India, it's widely associated with foods like milk, seafoods, vegetables, and its percentage of contamination varies from 1 to 12%. Listeria food poisoning occurs by consumption of food containing very high levels, more than 10 to the power of 7 per grams. So that is the number of organisms that are required to cause a disease. The incubation time is less than 24 hours, and the vomiting, diarrhea are the very common symptoms that you find with monocytogenes. The most important thing that we have to understand with monocytogenes is that it grows in the room low temperatures. That means a food which is kept in refrigerator also can have this low monocytogenes. The control is again essentially the hygiene part of it at various time points. The another pathogenic organism that we have to discuss in food safety are Campylobacter, and these are also gram-negative organisms. The Campylobacter jejuni is the most significant among the Campylobacter family. It is usually present in the gut of wide range of animals widely associated with the foods like poultry and seafoods. Quite a small number, that is about 500 to 600 cells, can cause illness. The appearance of symptoms will take 2 to 7 days, that is the incubation time. That means it can happen. You have a food, the symptoms may appear only after 2 or it may vary up to 7 days. Symptoms are diarrhea, sometimes bloody diarrhea also can be there. Cramps and vomiting are the very common symptoms of this disease. Contaminated water is the major vehicle for this transmission of Campylobacter infection, but it is very sensitive, high temperature and dry environment. It also presence of oxygen. In these conditions, it doesn't survive. The other important pathogenic organism is Staphylococcus aureus. It's a gram-positive, catalase-positive, cocal-shaped foodborne bacteria present as a part of normal microflora of humans and other animals. Even our own skin and nasal cavities will have a lot of Staphylococcus. This bacteria is widely associated in India in foods like koa, fish, meat and dairy products. Its percentage of contamination varies as high as 36% in Indian foods. The Staphylococcus food poisoning is called intoxication because it doesn't infect the person. The toxin that is produced by this particular bacteria will cause the disease. That's why it's called intoxication. And the toxin is heat stable. That's the reason why once toxin is formed in food, you can't do anything. It will cause a disease. And this Staphylococcus aureus results from the ingestion of foods containing immunologically distinct enterotoxins. There are different types of toxins produced by this organism. They are A, B, C1, C2, C3, D, E, F, G, H and I. These are different types of enterotoxins produced by different strains of Staphylococcus. Most of Staphylococcus aureus are caused due to the SA and SD enterotoxins. Other things are not generally found. The symptoms of this Staphylococcus aureus poisoning would be nausea, vomiting and diarrhea. The incubation time varies from 2 to 4 hours of ingestion of implicated food, and the minimum dose of this toxin would be 1 microgram or less. That means we are only talking in terms of the toxin present in the food, not the organism itself. Organism itself is not heat resistant. The organism can be easily killed, but the toxin, if it is produced, that is heat resistant. The other pathogenic organism is Yersinia enterocolitica. This is a gram-negative rod-shaped bacteria. This bacteria is having ability to grow at 4 degrees like Listeria monocytogenes. They can grow in the refrigeration conditions also. It causes foodborne disease called erisinosis. Symptoms of this disease are high fever, stomach pain, diarrhea. Sometimes vomiting also is observed. The incubation time is 2 to 3 days after you ingest the food. In India, this bacteria is widely associated with the foods like milk, buttermilk, lassi. The percentage of contamination varied from 5 to 59 in Indian foods. Yersinia enterocolitica produce heat stable enterotoxin, and in most of the cases yersiniosis are sporadic in nature. And the control of Yersinia enterocolitica is done by reducing the levels of contamination in the pre-processed food chain, destruction by thermal processing and adequate cooking at the household level, and prevention of recontamination. Shigella is the another pathogen in the bacteria that we are worried in food safety. It's a gram-negative non-motile Enterobacteriaceae member. There are 4 groups of Shigella differentiated by a combination of biological and serological characteristics such as dysentery, flexneri, boydii and soni. The infection caused by is termed as shigellosis. The organism is transmitted to humans both by food and water. The infectious dose for this particular organism is 1 to 10 to the power of 4 cells. The symptoms generally are diarrhea, bloody diarrhea and abdominal pain and fever. The common foods that are implicated for this infection is salads, lettuce, milk cheese, cooked rice. In India, the Shigella was isolated in foods like fish. Tamarind, its percentage varied from 1 to 4%. Infection of Shigella can be controlled by preventing contamination of salads, other foods eaten without cooking, and thorough cooking is another way of preventing this. Vibrio coli is the gram-negative, oxidase positive, facultative, anaerobic, motile, curved bacteria. It's both food and water-borne pathogens. Most important species of Vibrio are Vibrio cholera, Vibrio parahemolyticus, Vibrio vulnifocus. The foods which are associated with these organisms are meat, seafoods, and foodborne diseases caused by Vibrio species are fish, bread, vegetable curry and sandwiches. The cholera is caused by strains of Vibrio cholera, serogroup 010139. Serogroup 01 is having 2 biotypes and 3 serotypes. They are called classic even tar, or serotypes are Innova, Ogawa and Hikojima. The incubation period varies from 6 hours to 3 days, and the most prominent symptom of this disease would be passage of rice stools with water with a rapid loss of body fluids and minerals like salt and potassium. The excess fluid loss results in dehydration, hypertension, salt imbalance, and the other one is parahemolyticus, is also important pathogen that also causes diarrhea, abdominal cramps and nausea. The other organism that we are mostly Western countries are worried about, but in India we do not have much enough issue, but still we should understand this. When Clostridium botulinum, it's a gram-positive rod-shaped spore-forming anaerobic bacteria which produces foodborne disease called botulinum. The bacteria usually resides in canned foods. Clostridium botulinum produces toxin called neurotoxin. This is a very potent neurotoxin that is produced. It has different types of strains that produce A, B, E and rarely F, and type 3 rarely causes illness to humans. The botulinal toxin injection results in muscle paralysis. The early symptoms are mainly nausea, vomiting followed by neurological symptoms such as double vision, fixed dilated pupils, speech impediment, difficulty in swallowing. The incubation varies from 4 to 8 hours, and it can be as high as 36 hours also. The other Clostridium member that causes food poisoning is Clostridium perfringens. This is a gram-positive rod-shaped anaerobic bacteria and formerly known as C. welchi. It produces toxins from A, B, C, D, E and are responsible for serious diseases in humans such as gas gangrene and enterotoxemiasis. Symptoms are mainly abdominal pain, flatulence, diarrhea, nausea and vomiting. The infectious dose can be from 10 to the power of 6 to 10 to the power of 8 cells. The incubation time varies from 8 to 16 hours, and meat products are generally the carriers of this organism. Bacillus cereus is a very common organism that we found in India in most of the foods, and it is a gram-positive rod-shaped facultative anaerobic bacteria. This bacteria is usually associated with foods like milk, meat, cooked, uncooked rice, and its percentage of contamination varied from 5 to 50. It causes both infection and intoxication because some of these bacteria produce toxins also. They are called emitting diarrhenic toxins. The emitting toxin produced in foods, symptoms of that toxin would be vomiting, nausea and rarely diarrhea. The other one, diarrhenic toxin produced by in the gut tract, the symptoms are abdominal pain, diarrhea, but rarely vomiting and nausea. The emetic infective dose for emetic toxin would be 10 to the power of 5 to 10 to the power of 8 cells per gram of food where the food toxin is preformed in the food. In case of diarrhenic syndrome, the effective dose is 10 to the power of 5 to 10 to the power of 10 cells. Now we come to the other type of biological hazards, that is called virus. There are 3 different types of viruses that we are concerned with food. The most common one would be Norwalk-like virus, then rotavirus, and hepatitis A and E virus. So these are all generally associated. Hepatitis A and E virus, these are considered to be both food and waterborne pathogens. Acute hepatitis caused by HIV is self-limited and infrequently causes disease that results in death. The infection begins with mild illness characterized by malaise, low-grade fever, nausea and headache. The severe symptoms include vomiting, diarrhea and arthritis. These symptoms are accompanied by jaundice or illness, usually lasts to 2 to 4 weeks. The incubation period for HIV infection is about 28 days, but it can range from 2 to 6 weeks. Hepatitis E virus is primarily associated with the ingestion of contaminated drinking water. The incubation period varies from 22 to 60 days. It is a very long incubation time. The symptoms of HIV infection are nausea, dark urine, abdominal pain, vomiting, joint pain, rash and diarrhea. Quality of water supply is very important in prevention and control of these viral infections. Now we try to understand what is the Norwalk-like virus. Actually, Norwalk is a place in USA, since that was first that disease was observed in that particular community. That's why they have kept that name Norwalk, but now it's called norovirus. They are most common cause of foodborne viral illnesses. The virus transmits to human beings through contaminated food or water. Group of virus can occur in vomit as well as feces, and hence may spread through aerosol droplets also. Shellfish have been responsible for very large community outbreaks. Norwalk group of viruses are most frequently reported causes of foodborne viral illnesses in UK. The other one which causes almost 50% of under 5 diarrheal diseases is rotavirus. Contamination of rotavirus occurs through the use of fecal waters used as fertilizers or sewage effluents or irrigation of food crops, washing of fruits and vegetables with contaminated water, preparation or handling of food through rotavirus contaminated hands. The incubation period for this virus is around 1 to 3 days. The symptoms include vomiting, fever, respiratory illness. The other biological hazards include these protozoan parasites. These are Giardia, Cristosporidium, Cyclospora. These are the 3 different types of protozoan parasites that are generally found in both human and non-human hosts. Cryptosporidium causes cryptosporidiosis. The symptoms of this Cryptosporidium include the flu-like illness, tinea saginata and tinea solium. These are 2 known causes of tiniasis in humans. It is more common in human and domestic animals which live together under poor sanitary conditions, and undercooked beef or pork is generally contaminated with these things. Abdominal pain, nausea are the most common symptoms of the tiniasis, and cystic sarcosis is a systemic disease caused by the invasion of tissues by the infective cysticerci of staph forms. Tinea solium is also causes neurocystic sarcosis. Epilepsy is the most common sign of neurocystic sarcosis. The other helminthic infections will be Trichinella. It's a parasitic worm, causes a foodborne disease called trichinellosis. The mode of infection is from ingestion of meat harboring the larvae of parasite. It consists of elongated, cylindrical, colorless, fluid-filled body. It's transmitted through epidemiological cycles. The domestic and sylvastic. Domestic cycle consists of cyclic transmission of Trichinella from pig to pig, pig to rat, or rat to pig, and pig to humans. The other biological hazards include the fungi. These are some of the fungi which are toxigenic, and they produce toxins, and they are called mycotoxins. Most important, there are around 400 different types of mycotoxins are produced, but among them only 15 are generally concerned with food safety. The one is Aspergillus produces, that is called aflatoxins. It produces aflatoxin A, B1, B2, G1 and G2, and among them aflatoxin B1 is the most toxic among all other aflatoxin because it causes cancer. And the acrotoxin is another mycotoxin produced by Penicillium aspergillus species, and this is generally found in coffee, and they also have 3 different types of toxins. They are called acrotoxin A, B and C, and Aspergillus acraceus is the contaminant of wide range of commodities such as coffee, beer and wine. Acrotoxin A is considered as a potent nephrotoxin, and fumonis is another mycotoxin that is best suited among the fumonisins and produced by Fusarium molliforme. The fumonisins are mainly widespread in the maize and maize products. The last one is algal food poisoning. These are the marine algae which produce in seafoods. They include dinoflagellates which produce ciguatoxins associated with the major shellfish poisoning syndromes like paralytic shellfish poisoning, amnesic shellfish poisoning and neurotoxic shellfish poisoning. These are all actually fishes. They actually feed on this toxic marine algae, and that's how the fish will become toxic. The 4 important toxin producing genera of algae belong to dinoflagellates. They are Alexandrium, Gymnodinium, Dinophysis and Pronocentra. The concentration of as low as 200 cells per ml may produce toxic shellfish. Outbreaks of paralytic shellfish poisoning have been occurring regularly throughout the world. The toxins produced by Gymnodinium and Tychodiscus brevis causes neurotoxic shellfish poisoning. So, how do we control? We just discussed different types of biological hazards that can come into food from various sources, and the health effects also we have seen. But the most important thing for the control of these biological hazards is actually the ensuring food safety throughout the food chain, and most important would be the HACCP approach that is ideally suited for biological hazards control. Yeah, to summarize the module, what we have learned today is what are the biological hazards, because biological hazards are very important food safety concerns. That's the reason why we try to understand what are the biological hazards. They are generally bacteria, virus, fungi, algae and parasites. And among the bacteria, the most important things that we have learned today is the Salmonella, Staphylococcus aureus, Bacillus cereus, Listeria monocytogenes, Campylobacter, and among the viruses which are of concern would be Norwalk virus, rotavirus and hepatitis A and E. And coming to the toxigenic fungi that we have learned today is there are more than 400 different types of toxins are produced by these fungi, but among them hardly 12 to 15 of them are of food safety concern. Among them, the most important would be aflatoxin and some of the toxins like pathogen, acrotoxin. And among the parasites that we are supposed to be worried about will be Tinea solium and Trichinella and Tinea saginata. And coming to the algae, which actually some of the marine algae which produce toxins, if they are consumed by the fishes, then the fish actually accumulate these toxins in them, and the people who consume this type of toxic fish will have a food poisoning. That is another type of biological hazard that we have seen. And finally, the most important thing to prevent this type of biological hazards in foods would be to go by the HACCP approach from the farm to fork methodology.